Okay, so thank you, everybody. Actually, I work at the Weizmann Institute in Israel, but I'm also a clinical geneticist. So this sort of conference where the patients and the physicians and the scientists all come together is like a dream come true, especially when you come from Israel. So some of the talks today and on the previous days spoke about P53 and touched on metabolism. I'm going to do the opposite. I'm going to speak about cancer metabolism and touch on P53, but I hope that during the break you got all your metabolism going and that you're all set up for it. Okay, so most of you, the students, I don't know, maybe some of the children here that study biology, when they see this slide, it's really scary. But I want to try to convince you that it's actually very beautiful. Just by looking at the name and all the junctions here, you can realize how important metabolism is to almost any cellular process, disease, gene, anything you're working on will have to do with energy, with carbohydrates, with nucleotide. So it's very relevant to all of you. The second thing that jumps into mind is that basically this is a network. So everything is connected. This means that if we have a change or an aberration in one of the metabolic pathways, we will be able to see the consequences on the other metabolic pathways. So our thread that we use to untangle all the metabolic changes that we look for in different diseases is by looking at the urea cycle. So what is the urea cycle? The complete urea cycle occurs only in the liver, where ammonia that comes from the breakdown of protein and from the food is converted to urea that is secreted in our urine. There are patients that have germline mutations, not in P53, but in the urea cycle genes. And these patients suffer from what we call urea cycle disorders. And they have very high levels of ammonia that leads to neurotoxicity. In fact, in addition to having hyperammonemia, these patients also have very severe systemic manifestations because of the function of these enzymes outside the liver, outside the urea cycle. Most of them have cognitive delays, they have liver disease, and they have seizures. So the first enzyme that caught our attention was ASS1, and patients that have deficiency in ASS1, germline deficiency, have elevation of the upstream metabolite called citrulline, and this is why their disease is called citrulline. So because of the block, because ASS1 is not functioning, they have accumulation of the upstream metabolite, which is called citrin, and they have a deficiency of the downstream metabolite, which is called arginine. So as a physician, I had to take care of these patients, and they're very hard to metabolically control. And so it was very intriguing when papers started to come out showing that ASS1, and here you can see a northern blot, for some reason, in multiple and very diverse type of cancers, like melanoma, liver cancer, pancreatic cancer, very different cancer of origin. In all these cancers, for some reason, ASS1 was silenced, and this has clinical consequences, as you can see that patients, again, with very different types of cancers, when ASS1 was silenced in their cancer, they had worse prognosis, they had increased metastasis. So it was very intriguing to try to figure out what could be the metabolic benefit for ASS downregulation in cancer, because obviously it has to be something very robust since it involves so many different types of cancers. So outside the liver, ASS1 participate in another cycle to generate arginine, and most of the studies that looked at ASS expression in cancer thought that it has to do with arginine, because arginine is a metabolic nexus for multiple metabolites that are essential for cell proliferation, growth, survival, like proline, polyamines. We thought that when ASS1 is lost, we can take a different approach, and instead of looking at the downstream metabolites, maybe we need to look at the upstream metabolites, which are basically ASS1 substrates, aspartate and citrulline. So when you look at aspartate, cytosolic aspartate, and also glutamine, that is rich with nitrogen, are used by a cytosolic enzyme called CAD. And CAD is the bottleneck for the synthesis of pyrimidine synthesis. And if you look at the pyrimidine ring, most of the nitrogen and the carbons in this ring comes from aspartate and glutamine. So the hypothesis was that maybe when ASS1 is downregulated, aspartate availability for CAD increases, we have more pyrimidine synthesis, and we have increased proliferation. So why did I like this hypothesis? Basically because of the Warburg effect that was found almost 100 years ago, where he found that in cancers we have increase in glycolysis and decrease in oxidative phosphorylation, even though glycolysis is much less efficient in generating ATP and energy as compared to oxidative phosphorylation. And in the studies that follow, what was found is that basically we have increased glycolysis in cancers in order to supply the substrate that proliferating cells need. For example, they need fatty acids to generate membranes, they need to synthesize DNA and RNA to support the proliferation. And we thought that maybe our rewiring that we're documenting fits right in here, where ASS downregulation basically shifts aspartate from the arginine pathway to generate more DNA and RNA. So the way we do it in my lab to try to trace the fate of glutamine and aspartate, we use labeled carbons, and we basically trace them, and we look at the fate of this, for example, aspartate, that now its mass changed because 4 of its carbons are labeled, and we want to see how much of this aspartate goes into pyrimidine as compared to, for example, arginine. So just to remind you, our hypothesis, when ASS1 is downregulated, more aspartate, CAD is activated, pyrimidine synthesis is upregulated. In order to prove that, we downregulated ASS in some cancers, we overexpressed it in other cancers, and in both cases we saw that when we downregulate it, we increase aspartate availability, we increase cell proliferation, and when we overexpress it, we restrict proliferation. And this is important because this feature could be rescued by pyrimidine. So we have here a gene that when it's overexpressed, it restricts tumor growth, and it has to do with nucleotide availability for cell cycle. So naturally, we thought that maybe this could also be related to p53. And indeed, when we looked at ASS1 and we tried to analyze p53 binding site, we thought it has multiple binding sites. And when we induced p53 expression, we could see that when p53 is upregulated, also ASS1 is upregulated. So basically, what this suggested to us is that while in cancer, ASS1 is downregulated, we have high CAD activity, high pyrimidine synthesis, maybe when there is some kind of stress and p53 is upregulated, ASS1 will be upregulated as well. Pyrimidine synthesis, pyrimidine availability will be decreased. And what we're seeing is that this pushes the cells into cell cycle arrest. So with the help of Eitan Rupin, from the NCI, we looked at the TCGA database, and indeed we saw that in those cancers with mutated p53, which you see here in the red bars, there's upregulation of CAD, suggesting that maybe p53 promotes this rewiring and increases the metabolic availability when it's lost for increase in pyrimidine synthesis. So if a gene as important as p53 is involved, it led us to think that maybe this is relevant to other urea cycle genes and not just to ASS1. So one of the things we do in my lab, and basically here I think is like a great demonstration for this concept, is that we look at children that have germline mutations in rare metabolic disorders, just like the Li-Fraumeni patients, to try to understand what are the consequences of this specific changes in this specific genes to cancer, because in cancer, there's so many other multiple changes that occur simultaneously that if we're able to use a clean system, not generate a fish knockout or a mouse knockout, but look at this patient who was born with this specific change in this specific gene, maybe we will be able to isolate the direct from the secondary consequences of changes in this gene in the cancer. And another important reason to use this patient, any kind of germline mutation patients, is that our target eventually is to help cure cancer in patients. Nobody wants to, we want to cure cancer in mice, but we care less about it. So, just to show you an example, we took biopsies of fibroblasts from control and from different UC urea cycle patients. And as you can see here, this is just crystal violet staining. The stronger the staining, the higher the proliferation. You can see that in patients with urea cycle disorder, okay, no cancer, just by growing their fibroblasts, they have much more increased proliferation. And in all these cases, this increased proliferation was associated with CAD activation. So now we want to take it to the next step and ask whether this is relevant to cancer. So, as I told you, we showed that in the urea cycle genes, ASS1 increases aspartate availability for CAD. A very good friend of mine at UT Southwest, Ralph Debredinis, showed that another longer-named metabolite, carbamoyl phosphate, is also upregulated during specific type of lung cancer, and it increases CAD activity. And of course, glutamine and bicarbonate are also used by CAD. So this led us to think that maybe it's not just this gene or that gene that are abnormal in cancer. Maybe it's the whole pathway. Maybe the whole urea cycle pathway is rewired during cancer to increase the availability of aspartate, carbamoyl phosphate, and glutamine, all for the synthesis of pyrimidine. And maybe what we're talking about is a global dysregulation of the whole metabolic pathway. So we came up with what we call a UCD score. And why is it important? Because it's not just a complete loss of function of the urea cycle. For example, if we look at carbamoyl phosphate, in order for this metabolite to be elevated, we can either upregulate CPS1 or downregulate the enzyme that uses it, which is OTC. I know it's just names, but the principle is what is important. So we came up with this score, and then again, with the help of Eitan Rupin, we analyzed the whole TCGA data set for changes in expression of at least one gene in the direction that we want to increase the flux towards pyrimidine synthesis. And as you can see here, in multiple, and again, very different types of cancer, 2 or more of these genes change in the right direction, and the higher the UCD score we have, the higher CAD activation is. And when we had the data, for example, here it's for melanoma progression, when you start with normal melanocytes, moving on to nevus, primary melanoma, and metastatic, you see that UCD score elevates and correlates with it significantly. And again, what is important is that this correlates with survival, so patients that have this specific UCD rewiring in their cancer have a worse prognosis. So, can we use this for therapy, for monitoring? What I told you was that when we have this UCD score, we have increase in proliferation. But maybe we also have something else. Maybe when we have this huge increase in pyrimidine synthesis, maybe what we're generating in addition is a nucleotide imbalance because we have so many more pyrimidines in compared to purines. So we induce specific urea cycle perturbations in specific cancer cells, and in all the cases we could show that once we induce this perturbation, we are disturbing the 1-to-1 ratio between pyrimidines and purines. So pyrimidines, it's easy to remember, it's see you tomorrow, it's CU and T. And every pyrimidines, you know, on the DNA binds a purine, so we have like a 1-to-1 ratio. And in all the cases when we perturb one of the urea cycle genes, we got a change in this ratio, which was favoring pyrimidine. And why is this important? Because several papers showed that when we have nucleotide imbalance, we have insertion of mutation, because the increased availability of pyrimidines is so much more than the purines that the polymer is just inserted. And what we have is that in addition to proliferation, basically rewiring of the urea cycle can promote mutagenesis. So we analyzed, we sequenced all the cell lines that we had with the perturbation, and in all the cases, you can see here in the red bar, we had much higher mutations. But I want to emphasize that it's not just more mutations, it's very specific mutation where purine is replaced by pyrimidines. So we wanted to see whether this is true, not just in a cell line, but in a more global way of looking at it. And again, with the help of Eitan Rupin, we looked at the DNA, RNA, and protein of in the TCGA datasets. And in all the cases, we could show that when the urea cycle is dysregulated, again, you can see it in the red bars, we have increased in this specific pattern of mutations from purine to pyrimidines, and you can trace it from the DNA to the RNA to the protein. So basically what I'm saying is that this rewiring of the urea cycle leaves us with a genomic signature. So, as I said, we always want to see whether this has therapeutic implications for therapy, for detection. And in the recent years, immune checkpoint therapy receives much attention, and one of the criteria to give immune checkpoint therapy is because of the increase in the mutation. You have more mutations, you have more neopeptides, and you increase the response to the immune system. So we wanted to see if our signature also affects immunotherapy. And so we first analyzed data sets, published data sets of melanoma patients, and we could show that these patients that were treated either with anti-PD-1 or with anti-CTLA-4, that are both types of immune checkpoint therapy, the one that responded better had higher UCD score. And then we wanted to repeat the experiment, so we generated the UC perturbation in syngeneic mice. And you can see that when we gave the mice, the control mice, anti-PD-1, they responded, but not that much better. The urea cycle perturbation caused larger tumor, but the response was very significant. And I think instead of looking at the graph, it's much more impressive to look at each of these mice. These are just 5 examples in each group. When you give the control cancers anti-PD-1, the response is all over. Some respond better, some respond less. But when you look at the ones with urea cycle perturbation, almost all of them immediately responded. And this was associated with an increased infiltration of cytotoxic T cells. So next, I want to try to convince you that it's also important for detection, maybe early detection, because maybe the whole nitrogen homeostasis changes in cancer, because as I told you, the urea cycle main purpose is to get rid of excess nitrogen. And maybe in cancer, this the balance between urea secretion and pyrimidine synthesis changes. So basically what we do is look at the trash can and tell you what you ate. So our trash can is the urine, we look in it, and we try to figure out what happens to it when you have cancer. So if in normal physiology we have homeostasis because between what we excrete as urea and what we utilize for amino acids and nucleic acids, basically in cancer we expect this to shift. And indeed, when we analyzed human patients with prostate cancer and we looked at their urine, we found an increase in the secretion of nucleic acids. And this is one of my favorite figures, but I won't take too much of the time. When I analyzed the data from pediatric patients on the day of admission, on the day they were diagnosed with cancer, you can see here the case studies stratified for age and gender, but in almost all the cases, at least between age 2 and 18, these children had very low levels of urea, which usually doesn't ring any bell to physicians because we're more concerned about high levels of urea. So in summary, what I told you is that while in normal state, excess nitrogen goes through the urea cycle and is secreted, when we have cancer, there is no such thing as too much nitrogen. Cancer uses it all, proliferates, synthesizes pyrimidine and amino acids. It means the patient has a worse prognosis, but it also means he might respond better to immune therapy. And I'm trying to also convince you that it's important to look at cancer catabolism and not just think about its anabolism, even though cancer is an anabolic disease. So, just to summarize it all, I hope I convince you that metabolism, it's important, it's relevant, it's connected, so it gives you lots of hints. When you see an alteration, think whether it's an adaptation or cause. Use rare disorders. I think in this forum it's kind of obvious. Think about the flux and not just specific and anecdotal changes in metabolites. And I just want to say some thought-provocative idea that maybe when we lose P53 and we induce all these urea cycle changes, maybe we change nitrogen composition, and maybe this would allow us to monitor cancer, progression, detection, and maybe even increase the response to immune therapy. So, thank you.
That's it. No, a question? No, go. Question.
I have a question. Yeah.
Sorry, way up there,
David.
Sharon.
Oh.
Or who? Yeah.
You wanted to ask a question?
Yeah, sorry, I just turned it on. I've always wondered this. It shows some really interesting data about changes in the pathways. So, do patients with inborn errors of metabolism have a different cancer risk than other people? Has that been studied?
So, it's a group of multiple genes. Yes, some of them definitely have higher risk, like FH mutations, SDH mutations, they have higher risk. Some of them so far didn't live long enough for us to know. But yes, the answer is yes. You just have to know which specific genes increase the risk.
There's another.
Just one question here.
Sorry.
So with the P53 regulation of the urea cycle enzyme, so I wasn't sure. I wasn't sure if I missed it, but this is in the P53 null state, or was it mutate? Was this with like LFS mutations, for example?
So in normal physiology, anytime we tried several conditions, several different stress to induce, every time we induced P53, not in cancer, we upregulated ASS1. So we think, since it's like the genome guardian, one of the ways that P53 guards our genome is that when you have a problem synthesizing enough nucleotides, it would generate cell cycle arrest, also through upregulating ASS1 and depleting the metabolites that you need in order to support the nucleotide synthesis. Clear? Yes.
Very quick.
Very nice talk. So I'm just wondering, for the samples that you would need to understand the metabolism, how far is it from, say, collecting a large enough sample size?
So that's exactly what we're doing now. Because we want to see, because what we have is from the day of admission. I thought the urea would be high because the patient is so sick, but we were very surprised. He had like a urea of 2 or 3, very, very low levels. But we don't know if it's because it's the extreme time point. We need to see whether it can be a marker of early detection. But we know nitrogen is involved because it's part of the reason for cachectic patients. So we think it definitely has a. Thank you.
Thanks, Ayelet.